Good afternoon. I'd like to thank the organizers for inviting me to give this talk. My name is Joel Kostka, and I'm professor and associate chair for research at the Georgia Institute of Technology in Atlanta, Georgia. And in the aftermath of the Deepwater Horizon oil spill, I want to talk about how genomics and metagenomics revolutionized our ability to understand the role of microorganisms in oil spills and the response to oil spills. Of course, I can't summarize a decade of research in just a few minutes, so I hope that you'll look at our previous publications and also the publication that I've written for this conference to look into more detail on our discoveries. Some key publications, though. I co-chaired the legacy synthesis for the Gulf of Mexico Research Initiative in the area of microbial genomics, together with Mandy Joy and Rita Colwell. Last year, we published this colloquium report written by international experts on this topic. And then I also published a summary of our discoveries in the American Geophysical Union transactions. So I can't possibly talk about all of the aspects of the science of the Deepwater Horizon oil spill. I want to remind you that this was the world's largest accidental release of oil into the oceans, and much of that oil that was leaked into the deep ocean over a 3-month period roughly ended up in the coastal ocean and impacting coastal ecosystems, such as beaches that I'll be talking about. So why are we studying microorganisms and their role in oil spills? Well, for one reason, hydrocarbon degrading bacteria are ubiquitous, they're highly diverse, and they've adapted to consuming oil over millions of years of evolution. And biodegradation by microorganisms is often the fate of much of the discharged oil during an oil spill. So we're on the same page. I want to talk a little bit about biodegradation. And microbial biodegradation is a respiration process. That is, petroleum is a great food source for microorganisms, and it is degraded during respiration. Petroleum is then oxidized and decomposed, and oxygen is concomitantly respired to carbon dioxide and water, and then also to a lot of intermediates or daughter products, which may have different chemistries and toxicities than the original oil mixture. Just like any biological process, biodegradation and the organisms that do it need major nutrients such as nitrogen and phosphorus. So oxygen and oxygen and nutrients are master variables controlling this process. I also wanted to mention too that for those of you that may not be familiar with microbiology, that it's not just the microbes that degrade the oil that are important. It's really the whole food web, the whole microbial food web, and then the whole food web in general that impacts the fate of the oil. So in this cartoon here, the red microbes are the ones that eat the oil. We also have microbes that produce biosurfactants. They may chemically disperse the oil and facilitate the biodegradation. There are microbes that graze on bacteria. We've learned in the past decade or so that viruses also contributed to bacterial mortality. So all of these elements of the food web will transfer oil and impact the fate of the oil, these microbial interaction networks. I also want to make you aware of the fact that Metagenomics Technologies have, as I said before, really revolutionized our capability for studying microbes in nature. And this was the Deepwater Horizon discharge was the first major oil spill for which we had Metagenomics Technologies to study microbial communities in the environment, for which they were widely available. And so this allowed for the rapid, high throughput profiling of microorganisms in the Gulf of Mexico. What I want to spend most of my time talking about today is to give you a case study of the fate of the oil on Pensacola Beach, um, near Pensacola, of course. And this slide shows the extent of the oil spill in summer of 2010. This study was very much a team effort between myself and Kostas Konstantinidis at Georgia Tech and Marcus Hoodle at Florida State University. And this slide shows you sort of a visual history of oil on Pensacola Beach. Oil came ashore late in June of 2010. It's a very warm time of the year in Pensacola, and so you can see oil was pooling on the surface of the beach after it came ashore, and then it filtered down into the sands over the subsequent weeks until in late July you see that the oil had been buried a couple feet down in the sand column. Also in October of that same year, you see these oil layers present in the sand. In October, it was decided that BP contractors would come in and do a so-called deep cleaning, which was a physical cleaning where they brought in big road machines that dug up the contaminated sand, pulverized it, sieved it, mixed it with clean sand, and then dumped it back onto the beach. And so what you saw is that mixed the oil contamination over larger areas in the beach sand column. By April then of 2011, we can see that much of the oil was already biodegraded by indigenous microbial communities. And if you're interested, I hope you will look at some of our publications. This is the lower right-hand corner shows a lot of our publications from the last 10 years of studying this. This shows our people out there sampling in October of 2010. I just wanted to show you how we took samples. So we dug trenches to expose the beach face, and we sampled laterally through these oil sand layers. We also took cores, sand cores, through the beach to study these oil layers. What we found was that petroleum hydrocarbons were degraded relatively rapidly in these beach sands, and the microbial communities recovered after about 1 year from when oil came ashore. So Gulf beaches are very warm, and also oxygen penetrates down into the sand because it's permeable. And so this is an ideal environment for microbial growth and metabolism. And what we see, if you look in this right-hand panel here, is the colors are proportional to the oil concentration of aliphatics, aromatics, and total petroleum hydrocarbons from summer of 2010 to summer of 2011. And so what you can see is that the oil was filtered and mixed down into the sand column, as I just showed you in the pictures. And partially this was due to 2 tropical storms that happened in that summer that helped mixing the sand down into the beach column. And then rapidly over the ensuing months, you see that the oil concentrations were depleted through biodegradation, such that after 1 year, the oil was degraded to background levels. This oil also stimulated a huge bacterial bloom and a decline in microbial diversity. So what we think happens here is that when oil hits these ecosystems, that it selects for oil-degrading bacteria. They outcompete the other microbes that are present, and so oil degraders dominate the microbial community. So here you see a core that we collected during summer of 2010, and what you find in this oil layer is that the abundance of microorganisms is orders of magnitude higher than in the surrounding beach sand, and a large portion of the microbial communities are microorganisms that are capable of oil degradation. If you look in this bottom panel here, the bubble size is proportional to the abundance of microorganisms. So larger bubble size means larger concentrations of microorganisms or abundance of microorganisms. So microbial abundance increased by orders of magnitude proportionally to when oil was present. The black indicates where the oil was. And so just like we saw oil moving down into the beach sand column, we see that microbial abundance was moving down into the beach column following the oil. And Then by the end of the 2010 and in early 2011, you can see that the microbial abundance had returned more or less to normal. And the red line indicates that the taxonomic diversity of types of microorganisms went down when oil was present and then returned back to close to baseline in late 2010, early 2011. What we also observed was a succession of different microbial groups over time. And what we collected the largest metagenomic time series that was collected ever, I think, in oil-contaminated ecosystems. And what we found was certain microbial populations increased in relative abundance early on after oil hit the beach. And those were the microbes that used the more labile parts of the oil mixture, the alkanes, for example. And then later on, we saw different microbial populations that were coming up as the more recalcitrant compounds, such as aromatics, were degraded. And then we also saw some microbial populations decline in relative abundance and were inhibited by the oil. These were the organisms that don't eat oil, that are what we call chemolithoautotrophic organisms that are part of the nitrogen cycle. So certain microbial services were impacted by the oil. We were then able to use metagenomics to actually look at what microbial pathways and metabolic pathways were operative in the beach sands over time using this metagenomic time series. And we saw a very nice relationship between the abundance of genes for, say, alkane degradation or aromatics degradation with time and the presence of the oil and concentration of the oil. And then we could use this metagenomic time series to focus in on certain microbes that we found that were really important. So by studying their genomes, we could discover which microbes were important. So that's what you see in this lower panel here. The black indicates a certain microbial population that was up to 30% of the community in oil beach sands at Pensacola. By looking at its genome, we could tell that this organism not only degraded oil, but also was capable of fixing nitrogen, essentially creating its own nutrient source. And so we used that information from the metagenomics to actually bring this new species of microbe into culture, which we named Macondomonas or Macondo oil diazotrophica. And then when we looked at global gene sequence databases, we could see that this microbe was not only important in contaminated Pensacola beach sands, but also in contaminated coastal environments all over the world. It was present when oil contamination was present, but not in pristine coastal ecosystems. So we think it's a real important indicator for oil contamination. We then used all of the metagenomic data that we collected and expanded it all over the globe to create a curated, comprehensible, searchable database to document microbial populations in oil ecosystems. And we then documented those gene sequences along with underlying physicochemical data. We geocoded it with GIS, and we can use this to reveal the geographic distribution of oil degrading microorganisms all over the world. With this database, which contains over 2,000 metagenome assembled genomes, we were able to show that unlike what was thought previously, over 95% of the genomes that we recovered were unknown to science. So most of the oil degrading microbes and microbial species that we discovered were new to science and had not been studied before. And so this is kind of a family tree of hydrocarbon degrading bacteria, and we could see whole new branches of this tree that were associated with hydrocarbon degraders in the Gulf of Mexico. And lastly, in contrast to those diffuse oil particles that we studied in Pensacola Beach, we saw that tar balls degrade much more slowly than the diffuse oil contamination, although the microorganisms that we found in the tar balls were similar. In conclusion, then, we found a natural capacity of microbes in the Gulf, catalyzed bioremediation of petroleum hydrocarbons, that biodegradation was sufficient to remove diffuse oil contamination from beaches after 1 year, whereas larger aggregates persisted. We found novel species of microbes, novel metabolic pathways, and community dynamics using metagenomics technologies. And we found specific microbial groups and gene targets that show promise as sentinels for the mechanisms of hydrocarbon degradation and to elucidate the impacts of oil exposure on ecosystem health. Thank you for your attention.